Hi guys, this is Gaurav from Bilateral Anchor classes, and today we will watch at a new topic. We will see a new topic regarding the structure of a cell, right? So what we will be studying today is structure of a cell. So this is a beginner part, right? So for a particular beginner part, we need to understand what is basically a cell. A cell, we can say that we can define a particular cell by telling it as a structural and number 2 functional unit, structural and functional unit of a body or organism, right? So in case of a body or organism, this is the smallest unit, as we can find it that in case of matter, the smallest unit is an atom, right? In this particular case, we can find it that it is the structural unit and functional unit of a body of an organism. So how does it signifies that it's a structural unit or a functional unit? From these two things we can understand that there are two features of a particular cell, right? The first one is structural feature. Why it is known as a structural unit? Now we live in houses or buildings, or nowadays we can see that the houses or buildings are built by bricks, right? Now if we take suppose this is a house or building and this is a body. So building and a house building, a body or house building, how can we relate between these two by using a cell? Suppose in case of a body, we can see it is made up of a single cell, and in case of a house or building, we can say the primary unit. Now we are taking it as primary unit, right? That is the primary unit. Okay, so primary unit, this will be a brick in this case, this will be a primary unit too. And so lots of cells, when they accumulate with one another, they form a particular tissue, right? But in this particular case, what we can see that lots and lots of bricks, they form a particular wall. Now look how similar is this. A single cell, when it aggregates with too many cells which are structurally and functionally same, are known as tissue. A brick which is structurally and functionally obviously similar, it forms a wall, right? Now too many tissues form an organ. Maybe I am drawing in here liver. No, it doesn't require drawing. Okay, right? So in this case, a wall, 4 wall, 6 walls. 6 walls makes a room, and many organs form an organ system. And what we know that it was primary, this was secondary. Suppose this is tertiary and this is quaternary. Quaternary organ system, right? In this case, it will form a particular apartment. It will comprise of too many rooms, right? We know that it could be of 2 BHK or 3 BHK room, right? And in this case, it will be a particular organ that will lead to a particular organ system, and too many organ systems will form a body, and in here too many apartments or floors, maybe we can say it as a floor, we can have a building. So this is how the structural fundamentality of a cell works. First, it organizes tissue, too many cells organizes tissue, too many tissues organize an organ, too many organs organize an organ system, and then too many organ system forms a body, and it has a similar systems like brick, wall, room, apartment and building. So now let's move on that how a particular tissue would look like. But before that, we would like to approach on our next topic, that's types of cells, right? Mainly depending on the presence of nuclear material and different cell organelles, we can characterize a particular tissue in two types, right? A particular cell in two types. So cells we can divide it in prokaryotes, and number two we can take it as eukaryote. Now, what's the difference between this prokaryote and eukaryote? So, prokaryotic animals, organisms, right? Prokaryotic organisms, they actually lack a particular developed nucleus, and in case of eukaryotic organism, they will have one organized, well-developed nuclei. So, how is it? Suppose I am drawing bacteria in here, right? What would happen? After its outer layering, I will discuss it in later classes when we will be discussing about the prokaryotic cell structures. We will find that the nuclear material or nucleoid, it is known as nucleoid. Nucleoid will be floating in the cytoplasm in a free way. But in case of eukaryotes, we can see that the nuclear material or DNA will be floating inside a nuclear membrane, and this is our nuclear material. So that will be the basic difference. There are loads and loads of difference between these two cells, right? Now, another category we can have in here. So, prokaryotes have their unorganized nuclear material in the form of nucleoid, and nuclear membrane and nuclear material containing eukaryotic cell will be an organized form. They will be in organized form, right? So this will be the basic difference. I'm just giving you an overview for different kinds of exams. Actually, we prepare for NEET, and I taught for the NEET actually, and afterwards there are classes of JEE, and certain people ask me about the UPSC exams also. So we have to clear these chapters for our much more appreciation in the marks, right? So the next category, what we will be looking at, that these prokaryotes, on our further rotation, we will be looking this one in later scale, later classes, right? Now, in case of eukaryotes, we can see these two kinds of cells. There are mainly two kinds of cells. Number one, that is an animal cell, and another one is a plant cell, right? So animal cells, it will look like this one, having a definite nuclei or nucleus, having chromatids. I will discuss it in later classes. Chromatids. And in case of plant cells, we can see that in animal cell there will be cell membrane, but in case of plant cells, we will have both the cell membrane and the cell wall. We will have both the cell membrane and the cell wall. So in this particular section, we can see that plant cells will have a larger vacuole, In animal cells, the vacuoles will be smaller, right? The vacuoles will be smaller vacuoles. In case of animal cells, we will see that there is presence of Golgi body or Golgi apparatus. In this particular case, we will see that it will have some dictyosomes. Now these two differences will be dealt with. These two differences will be dealt in such a case that we can see in here that dictyosomes will be present in plant cells and Golgi body will be present in animal cells, right? Now these plant cells will have a plastid. As we know that plant can prepare their own food, that's why these will have their own plastids. But here no plastid will be present, right? And another thing is that the cell wall that what we have written in here, in certain kind of fungi cells we can see that it will have a cell wall made up of chitin, and this cell wall is mainly made up of three things, right? Number one, lignin. Number two, calcium or magnesium pectate. And number three, we will see that it will have cellulose, and there are certain things like suberin that will be known as suberin. So these will be the most distinct differentiation between plant cells and animal cells, right? Now we will next, in our next topic, we will see the basic structures of these two, right? So let's move on to our next diagram. That's it. That is the structure of a particular cell, right? So we will be taking the animal cell first, but because it is much more easier than the plant cells. Okay, so first let's discuss the structure of animal cell, and then we will move forward for the next structure, that is the plant cells, right?